about. Okay, so we are recording. I'll get. I'm just a little rusty, just shaking off the rust. That's all that is. Um, um, um so in the virology class, general virology, I don't know. I'm gonna say this incorrectly. Jalissa, is that correct? Jalissa.
Yeah.
Um, Great, Jalissa, calling out Dr. Adams, ready for the weekend. Like it. Um, so um, in general virology, which is after this class, I went straight in recording, but I always stop recording and then start it again is what I'm supposed to do, because if you post the video, even with a little clip of the song that you play at the beginning of class, then all of a sudden YouTube freaks out and they think you're like pirating like all these crazy things. And so um, I did that, and then I got like, it wasn't like an official warning, but it's like you might want to double check this. And then I was like, oh my gosh. And so then I had to like edit it. It's like I can't win sometimes. You can't win. But yeah. Anyway, I'm looking forward to acing this next week for recording. So back to Harold Amos. Dr. Harold Amos, he got at a when he was a kid, he got this biography on Louis Pasteur, and he confessed later in life that part of the reason he was really interested in microbiology and immunology is because Pasteur used goats as experimental animals, and he did not like their family goat, um, or like goat milk. And so like that actually was a random thing that like he was like, oh, like this guy uses goats for his research and I don't like my goats. So your inspiration for science can come from surprising places. He attended school at a time when it was segregated, and also ended up going to college, the Springfield College in Springfield, Massachusetts, which he was awarded a full academic scholarship for, which was very rare for African Americans at the time. And he graduated summa cum laude in 1941 with a major in biology, minor in chemistry. Could be like many of you. Um, then he was drafted into the Army, World War II, and also was part of the Normandy invasion. After the war, he attended graduate school at Harvard, ended up earning a master's and a PhD, and became the first African American to earn a doctoral degree from the Division of Medical Sciences at Harvard University. So, really a pioneer for um, African Americans. And he studied a number of things, including the herpes virus and using a chick embryo model to study viral uh, replication and agents that inhibit that. And then, as a faculty member at Harvard, he studied a wide range of topics, including how you can manipulate bacterial RNA to program protein synthesis in eukaryotic cells, and looked at how you can optimize insulin production, um, RNA metabolism, and really a diverse range of topics at the time. So really sort of had his hands in everything. And then at the age of 70, he retired. All right. So um, I don't know when people think about retirement, but 70 is getting up there. But his retirement transitioned him to becoming the first national director for the Harold Amos Medical Faculty Development Program. So it wasn't originally called this, but they named it after him because he worked as its director for over 2 decades. And their basic mission is working to increase the number of medical, dental, and nursing faculty from historically disadvantaged backgrounds. So, one of the most impressive things I learned about his life is that while he was a pioneer for racial equity and justice, as well as a brilliant scientist, he also remembered to pay it forward, which I think is so important, to give back to the communities that helped you grow, and to make sure that you help others follow in your footsteps and make even more progress. So that is Harold Amos. He has a very interesting life. I encourage you to Google him, check him out a little bit more, and learn about the wonderful things he did. With that, we're going to jump into our class slides, and this is just a reminder to go ahead and set up your iClickers. Almost all of you have by this point, and I apologize. Last class, I had not synced the iClickers with the Canvas page on my end, so I think that was causing a complication on your end. The rosters are all connected now, so everyone should be able to fully connect iClickers to the class account, and it should automatically be integrated into Canvas. Just in case there's any confusion on that still, I'm going to start the participation for iClickers on Tuesday, February 9th, but today is still a good trial run to make sure that you are all set on that end. And if you are still having difficulties, please reach out to me or attend the Student Success Hours tomorrow from 10 to 11. So that is, I think, all the announcements we have. We left off on the amazing, universally, I don't know what to say, applicable gram stain. The answer was dark purple. And we didn't quite have time to show the explanation slide, although most of you got it right. After that 1 minute stain with iodine, right, you're going to have those gram negative cells also be purple, as are the gram positive cells. So that is sort of the takeaway in terms of rationale. And also, if we remember, stain both gram positive and gram negative cells. We do not want to hurt anyone's feelings, okay? And again, the cell feelings chapter is later on in the course. I think it's chapter 27, but we will get there. So don't you worry. Oh, right. Yes. I think I've mentioned I am not perfect, but for those of you who are still holding out hope, let me fully crush it. I did make a mistake from last class, and I am always open to being corrected or, owning up when I am wrong. So I apologize. We had some really interesting class questions on evolution and which came first, the chicken or the egg, bacteria or eukarya, etc. And actually, I wanted to explain that I believe I was saying that archaea and bacteria are most closely related, when in fact I was misremembering, and it's actually eukarya and archaea have been posited or proposed to be most closely related relative to bacteria. But because I really do enjoy exploring science, and also I want to make sure I can answer your questions and dig deeper if I don't have the answers on hand. I did a little poking around. All right, so this is the phylogenetic tree of life. And so this model has been proposed most recently by Carl Woese, who did a lot of sequencing analysis of core sets of genes in these three domains. And if you're not familiar with phylogenetic trees, basically the different branches reflect the different clades of organisms that make up life as we know it. And the entire basis for this revolves around a last common ancestor, which I alluded to last time. And so that would be in the center here, where you sort of have this intersection of the last common ancestor. And then there's a split. There's a fundamental evolutionary split, and that allows different life forms, that reflects divergence of the different life forms. And so there's a split between bacteria and archaea and eukarya, right? But the split is really between bacteria and then these two groups. And then this, at a later time point, there is a split between archaea and eukarya. All right, so that's a model that has been proposed as recently as 2013. And then around 2013, there was a newer model that has also been proposed, which looks at the three-domain hypothesis. So that's what this previous model is called, the three-domain hypothesis. And then an eocyte hypothesis or a two-domain hypothesis. So basically, in the original hypothesis, you have bacteria as one domain, eukarya as another domain, and then archaea as the final domain. But more sequencing data has come out. Scientists are constantly evaluating hypotheses, collecting more data, reevaluating hypotheses, testing different models. And now another model posits that there are actually only two domains of life originally, bacteria and archaea, and that eukaryotes are actually derived from an archaeal clade. So there was an initial split, and bacteria and archaea were the two options, right? You're either blue or green, right? So you're those are the two color options, or those are the two life clade options. And then at a later point, eukarya emerged from a common archaeal species. Okay, and so some of the logic for this is that if we think about the eukaryotic cell, it has mitochondria, which have long been thought to have once been bacteria that were able to inhabit eukaryotic cells, and now they have lost their ability to be independent from eukaryotic cells, right? So the theory goes that there is a archaeal organism that engulfed a bacterial organism, and then a eukaryotic organism emerged. So that is an additional hypothesis that's out there. So I'm sorry if this was more detail than you wanted about evolution of microbes, but I really do take your questions seriously, and I'm committed to answering them as best I can. So if you would like more information, I encourage you to check out this paper. It goes into a great detail about all the sequencing information and analysis of these different models. And I am always happy to correct mistakes if I make them, and also answer your questions throughout the class. So, oh, great. Yeah, I forgot. I actually made a few more slides, but I'll just post these in the interest of time. But this sort of walks you through the key points there. All right. So I'm just going to check the chat if there are any quick questions. Looks like we're good. So great. And we will return to this topic later on in the course as well. So one of the important concepts that allow us to visualize bacteria has to do with excitation and emission. All right. And this is a process where we can use the principles of chemistry to our advantage. And so when we look at a specific molecule, we can use this concept of fluorescence to see either a specific component of a cell, the entire cell. It depends on what we're targeting and what our sample of interest is. The model that is presented here goes over how these electrons interface with energy that then gives off fluorescence. In the first step, you have a UV photon that is absorbed by an electron. All right, so you have electrons circling in orbitals, and this electron is then raised to an orbital of higher energy. And so you could think of the electron as like, hey, it's just having a chill day, but then all of a sudden it's like oh, I'm gonna have some coffee. And it's like, and it's like super excited. It's like, wow, like let's party, right? So it starts like dancing, whatever, right? The electron is gonna lose some of that energy, right? Because you're dancing, it has a bunch of heat, right? So it's losing that energy, that energy in the form of heat, and it's gonna like drop down just a little bit. It's still more excited than it once was, but it's gonna lose a little bit of that energy. After it's lost a little bit, the rest will be emitted as fluorescence. And this will be emitted as a um, wavelength that is longer um, and at lower energy than the initial wavelength that it was excited by. And once this fluorescence event happens, the electron returns to its normal chill level. This entire process is also reflected in this graph here, where you can see the relative intensity um, of the signal that is being um, measured, as well as the wavelength, with the shortest wavelength being on the left, and then it gets increasingly longer on the right. So you can see that the absorption or excitation wavelength is higher in terms of its energy and shorter in terms of its wavelength. And then later you have the emission wavelength, which is lower in terms of its energy and higher in terms of its wavelength. So hopefully that makes sense conceptually. But if not, please let me know so we can go over it further. And this basic principle of fluorescence is really important for the optics when we think of looking at microbial specimens via microscopy, right? We can use the principles of light, fluorescence, and also specific filters to carefully calibrate the excitation and emission spectra that we view. And the basic principle that guides this is the use of a light source that then passes through a very specific excitation filter, which allows a specific excitation wavelength on to our sample, right? And through on our objective lens. This signal is then reflected back to us via the fluorescent wavelength, which passes through a dichroic mirror, okay? And dichroic mirrors are a specific kind of mirror that reflect light below a certain wavelength, but transmit it that light that is above that wavelength, okay? And by using this setup, we are then able to view these objects that fluoresce or that we stain with fluorescent antibodies to then visualize specific components. And this second strategy of being able to visualize specific objects with fluorescent antibodies is made use through what we call fluorophores. And there are different variations of how you can incorporate a fluorophore into your sample, okay? One is often a antibody that is conjugated to a fluorophore, but there are also gene fusion reporters and a variety of other approaches that can be used. The important considerations to think about when staining a sample of interest is how you're going to determine the specificity of your fluorophore. And the chemical affinity, the labeled antibody approaches, DNA hybridization, and gene fusion reporters are all tools in your toolkit to approach your microscopy analysis and visualization. Here we're looking at an image where they've stained specifically the DNA origin as well as the septum of these B subtilis or Bacillus subtilis cells during sporulation. And this allows you to have enhanced detail of the samples that you're imaging. Here is some research from my own lab. A simple slide looking at a lung epithelial monolayer. And this also illustrates the use of fluorescent staining of a sample. Okay, so here at San Jose State, we can use DAPI, which binds DNA in our host cells, as well as phalloidin, which binds F-actin or filamentous actin around the periphery of the cells. And together, this enables us to have a close appreciation for the integrity of the membrane. We see that the cells are mostly confluent and in good health. If we were to infect them with bacteria, then we might see a different picture, and we could start to see the degradation of specific proteins or damage to the cells. We're now going to go ahead and do an eye clicker question. So go ahead and hopefully log in if you haven't done so already. The question is, in fluorescence microscopy, the specimen absorbs incident light and then re-emits it at a blank energy and thus a blank wavelength. Lower, shorter, lower, longer, higher, shorter, higher, longer, good, totally gnarly. All right, 45 seconds on the clock. Go ahead and take a stab at this. Remember, it's participation that counts. Technically, everyone's getting free participation this week, but don't let that stop you from participating now. The best time to participate is when, well, always. That's the best time to participate. Yeah. "15 seconds left. Don't miss out. This is for all the marbles." All right, we are reached 45 seconds. It's not really for all the marbles. It'd be pretty high stakes if it was. Can you imagine that? One question to determine your entire grade. Whew. I want to get that one wrong. Alrighty, we have. Okay, we have. Oh, uh-oh. Did that just disappear? Okay, well, there we go. Sorry. So we have 51% of the class saying lower, longer, and then 33% of the class saying higher, shorter, and then some sporadic guesses elsewhere. One person is committed to good and totally gnarly. This can give you the bad news now. That is not correct, although it did make me think of surfing with all these wave terms being thrown around. Does anyone want to give an explanation for how they voted in this iClicker question? Maybe try to convince some of your other classmates.
I said lower and longer because
Okay.
I feel like lower energy is more stable, so it makes sense for something to be re-emitted at that point. And I know that energy and wavelengths are inversely related, so the lower the energy, the longer the wavelength, and vice versa, I think.
Great. Thank you so much. I didn't see who was speaking. I apologize. Could you say your name?
Oh, yeah, Darina.
Darina, thank you very much. Do people agree with that? Do people want to counter that in terms of their input for this question? In the chat, Janie, if I'm not getting that right, please correct me, is saying the specimen emitted light that passes the mirror and a lower energy wavelength is longer. Okay, looks like we have good consensus. And in fact, that is correct. So good job in terms of the analysis and explanation there. If people have questions, please come to the student success hours. I'm happy to go over that concept in more detail. But you definitely want to make sure you are on the same page here, as there was a significant number that maybe missed a little bit of something, but that's totally okay. I always say resolve your confusion early and often, so when the exam comes, you'll be ready to crush it. Let's go ahead and move on to the next topic, which has to do with the cell wall and its outer layers. The cell wall itself is considered the most common structural support for microbes, although there are different variations depending on your microbe of interest on how that structural support is, or what it is comprised of. For the majority of the microbes we'll talk about, we will be thinking of cell walls as peptidoglycan or mirin. And this is a molecule that consists of long polymers and 2 disaccharides. We touched on this a little bit last time, called N-acetylglucosamine and N-acetylmuramic acid. And they bind to a peptide of 4 to 6 amino acids. These molecules form cross bridges that are able to connect their parallel glycan strands. And if you think back to the peptidoglycan and whether it is present in large or small amounts in gram-negative and gram-positive bacteria, you can hopefully start to integrate this into our broader understanding of the structural components of microbes. And man, I'm so bad at this. Which one has a lot of peptidoglycan? Is it gram negative? Is it gram positive? I'm totally blanking.
Gram positive.
It's gram positive. Carlos, are you sure? Are you sure it's gram positive?
That has a lot of peptidoglycan. I'm pretty sure if I remember correctly, because it's the darker one, because more peptidoglycan, more of the staining. That's what I can remember.
Perfect. Yeah, I can't fool this class. All right, keep it up. Don't let a questioning voice fool you. If you're confident, you're confident. Yes, gram positive have tons of peptidoglycan. And if you weren't sure, remember the negative sign for gram negative cells can also be a helpful reminder that there's a thin layer of peptidoglycan for the gram negative ones. Here we're looking a little closer at these cross bridges that form in between the peptidoglycan strands. And we can see a close-up of this diagram here, where we see these specific amino acids that are then being linked together between the glycan strands. And it's important to point out that because all bacteria have peptidoglycan, this makes for a very attractive target in terms of antibiotic therapy. And in fact, penicillin is able to target the transpeptidase that cross-links these amino acids to one another. What better way to destroy bacteria than take out their cell wall, right? This is similar to the dragon in Game of Thrones. It just sort of punches a hole in the wall in the north. And sorry if that's a spoiler at this point, but yeah, it's bad news if you're counting on your wall to protect you. Analogous to this is the antibiotic vancomycin, which actually is able to bind to the 2 terminal dealanine amino acids. And by binding to those 2, it prevents removal of the second dealanine. What this means is, once again, you do not have cross-bridge formation between your glycan strands. So both of these antibiotics function differently, but overall, the result is the same, and that you're preventing cross bridge formation. And thinking about mechanistic differences here is important because that ultimately means that bacteria need to evolve different ways of becoming resistant to these different antibiotics. Now, unfortunately for us, bacteria are not lazy, and they are actually quite resilient when it comes to developing antibiotic resistance. I'm sure many of us are familiar with the widespread cases of antibiotic resistance, the so-called superbugs, and multiple antibiotic resistant strains, MRSA, or multi-resistant Staphylococcus aureus is part of the joke of this comic here. And finding ways to limit antibiotic use and having it be selective as opposed to everyone take antibiotics all the time will be really important in helping us win this battle against antibiotic resistant bacteria. Because once we run out of antibiotics, we really will be in a very dangerous world where suddenly even very simple infections are no longer treatable because we're out of these therapies. The other important consideration to think about as we go over sort of fundamental aspects of bacterial cell structure and shape are the different cytoskeletal proteins that are revealed by these gene defects in different bacterial mutants. What we can see here is wild type cells and then bacterial cells lacking specific proteins. Several of these proteins have been identified in the research on how bacteria form their shapes or their specific structures. Here we're seeing 3 different examples: Staphylococcus, Escherichia coli, and Caulobacter crescentus. These 3 microbes have these shape determining proteins. Well, really all microbes that we are aware of have some kind of shape determining protein that is responsible for orienting their growth and specific morphology. FitzS forms a Z ring that you can see in the center here for spherical cells. MREB forms coils, and Cre-S or crescentin forms a polymer that is along the inner side of these crescent-shaped cells. Now, one thing that I wanted to briefly comment on is that I know we are all using some edition of microbiology and evolving science. For those of you using the latest edition, you will see certain things that maybe are minor details or other additions that are not on some of these slides. So for some of you using the 4th edition, you'll see often exactly what I'm showing, and some of you using the 3rd edition might see what I'm showing or not, maybe a slightly older version. Just so we're all on the same virtual page, I guess. I will definitely be moving back and forth between the different editions, and this is not to be confusing, but it is really out of equity. So I think it's really pretty outrageous that the newest edition of the book costs almost $200, but the 3rd edition is like $4 or $5. And so if you are concerned if, you know, oh, my book isn't showing this or it's showing something slightly differently, do not worry. My tests are exclusively based off the slides and the material we cover, not so much what is explicitly in the textbook, although a lot of times they are one and the same. But I just want to make sure that if you're, comparing what you see on the slide to your textbook and you see something that's slightly different, always know that the slide is the default option there, or is what you should default to. Don't be confused about the different variants in the book. So I will incorporate figures from all 3 editions, but it just is going to depend on the content that we're covering for the class. If you have questions about that, I will try to make a little bit of time at the end of class, just so there is no confusion on that point. All right, so a couple of core terms that we should all be familiar with here, hopefully already, but we have cytoplasm, right? So this is sort of like that gel-like network in the inside the cell itself. You have the membrane that encloses the cytoplasm, and you have the cell wall, which we have spent some time talking about, that covers the cell membrane. You have the nucleoid, which is this non-membrane bound area of the cytoplasm, right, containing the chromosome and its loops, or looped coils. And finally, not all bacteria have this, of course, but flagellum are these external helical filaments. They're the little engines that are allowing the microbes to motor along. If we now focus a little bit more on the idea of bacterial replication and microbial replication, it's important to think about the steps that the microbe must undergo to make this event happen, right? 1 becomes 2, and then 2 become a million given enough time and nutrients. Here we can see a microscopy image of the host cell being attacked by some E. coli. These E. coli are actually replicating on what are called actin pedestals. We'll get to this towards the end of the course, but these attaching and effacing E. coli are actually able to inject host cells with toxins and other proteins to help mediate this formation. You see, this is literally the definition of E. coli putting themselves on a pedestal. So that's it in action. And these cells have a strategy for replicating that follows some basic principles. First, let's talk a little bit how DNA is organized. If you remember from last class, we talked about how it could be very useful to have a nice compact genome, not a messy, disorganized one. And the E. coli nucleoid itself appears to have different clear regions that exclude the ribosomes and contain DNA strands. So you can see this nucleoid region here, right? And the ribosomes are present in other spaces. And so you have these loops or domains that each have DNA that is supercoiled by DNA binding proteins. You can see an individual loop here. And this is directly connected to how they mediate their transcription and translation events, okay? What we have is during the replication of DNA, you can have cell division occurring, but you also, depending on the environmental and regulatory signals that you're processing as a cell, you have the generation of RNA from the DNA, right? So I'm sure we're all at least somewhat familiar with this process. We'll be digging into it in more detail later on, but for now, this provides a basic overview of RNA polymerase moving along to generate the RNA from DNA, which can form these hairpin loops and is subsequently processed by ribosomes into proteins. This process is aided by transfer RNA or tRNA, which brings the amino acids to the ribosome. So if the ribosome is where the party is at, then tRNA shows up with some amino acids and then they make a protein. It's a micro party, so it's not necessarily the coolest, but depends on what protein you're making, I guess. And so, the translation process is actually tightly coupled to transcription, and this helps the microbes have a very rapid response to different environmental cues. So if all of a sudden a microbe finds itself in a increasingly warm environment, if the temperature is rapidly rising, then the response needs to be rapid. So different stress response proteins can help the bacteria survive before it's too late. If this takes days, then the bacteria aren't going to be alive, right? The bacteria need a response in seconds or minutes to make sure that they are going to be around past this intense environmental stress. When we think about the transcription and translation theme from a slightly broader perspective, here we're a little bit more zoomed out. We could see that the membrane proteins themselves are synthesized in association with this membrane, and it is aided by signal recognition particles, which are abbreviated SRP here. Oh, sorry, my mouse is being a little finicky today. And basically, this will bind to growing peptides from the ribosome and help facilitate the association with the membrane. Now we're going to go over some of the key steps in a dividing cell. So this, again, is going to be a overview of the process, and then we will take a deeper dive in a few weeks. So in the bacterial cells, there is a circular chromosome, and replication begins at what is called the origin of replication, or the ORI site, and that is indicated in green here. Next, you have the DNA start to unzip, and this is because you have the formation of a replosome shown here. So you have the DNA unzip, or the DNA helix unzip, and the origins will actually replicate, and then you have migration occur. And this DNA replication continues in a bi-directional manner, all right? So it's a little difficult to visualize, and I don't want you to stress out about it too much, but I'm just gonna sort of walk you through the key steps. Not all of them are shown here, but I still want you to try and at least conceptually wrap your mind around what is happening. So basically, if you look at these two replisomes, okay, these are molecular devices that are generating the DNA, right? So they're replicating the DNA, and they are essentially pulling the DNA through and then generating more DNA. Okay, so it's sort of, if you imagine that the DNA is just being threaded in, let me switch to a marker here. All right, so you have the DNA being threaded in and the replosome is moving along, then you have essentially the DNA replication process occurring as it goes through the replosome. So if you compare here to here, you see that the replosome has now pulled through a bunch of DNA and is moved all the way to here. So if you look at the distance between the replosome and the origin of replication right now, and then later on, you see that this distance has grown a lot because of all the DNA that has been pulled through. And so this unzipping process that occurs here is referred to as the replication fork, okay? This replication forks are sites of DNA synthesis by DNA polymerase, and there are a number of accessory proteins that also help with this process. The key factors that contribute to the, this process are the DNA polymerase and the accessory proteins, and together that is what is referred to as the replosome, and they're just showing this as this yellowish blob right now, but it is actually a more complex structure. So you have DNA being pulled through the replosome. This is where the replication fork exists. And then you have the generation of new DNA via this process. So the replication forks proceed outwards in both directions. And because the sites of the origins of replication are attached to the cell envelope, they actually move further apart. So if you look at where they're positioned now and where they're positioned earlier, you see that in fact, they're spreading apart. So they used to be best friends, and then they didn't keep in touch. They slowly grew further apart, and now they barely know each other. It's, really, really quite sad. It's a sad movie. But that's how life works. Cells have feelings. So you have these replication forks. You have the origins of replication growing further apart, and don't worry about these new ones right now. Just pretend they're not there. Eventually, these replisomes will reach the end of their journey. Okay, it's like they're graduating or something, and they will reach the termination site that is present on the DNA. And this will say, all right, you finished, congratulations. Now you can do this all over again at a new site of origin of replication, okay? At the same time, often you have new replication forks being created by additional replisomes. So this is a cyclical process. They're always, well, not always, but oftentimes you are constantly synthesizing new DNA with multiple strands, multiple replisomes. At a certain point, depending on other environmental signals, etc., you have septum formation that occurs. And this is tightly coordinated because you want to make sure that you have a complete copy of the DNA in your cell before you have separation. If not, then your cell will probably not survive. So at this point, you have new origin sites and you have new replisomes that are creating new DNA at the replication forks. So as this process continues, you will ultimately get division at the septum, and then this cycle begins anew. So I know that's a lot to sort of wrap your head around, but this is, again, sort of a survey of some of the fundamental themes that we'll be covering in this course. And they don't show all the steps here, and they definitely don't show them in exquisite detail. So basically try to focus on the big picture about what the key players are. So replisomes, sites of origins of replication, sites of termination, septum formation, and division. Don't get so hung up on which way is the DNA moving, where did these replisomes come from out of thin air, what happens if the sites of termination or the origins of replication aren't friends anymore. I mean, these are questions that we'll get to later in the course. But because I know that was a lot to process, are there any questions right now about what we've covered? And let me pull up the chat. All right, it looks like there are no immediate questions. So I'll ask a reflection one. You have these cells and they are dividing, they're replicating their DNA, but what are your thoughts on how these dividing cells would eat nutrients during this time? Any guesses or hypotheses? How would they eat nutrients if they're so busy replicating and dividing? And I can also put that in the chat.
Would it be through their environment?
Through their
Like,
environment?
yeah, like depending on where they're at. I mean, I'm sure the membrane is permeable, or like stuff can go through the membrane so they can take it in at the same time.
Thanks for sharing, Carlos. Yeah, so is it through their environment? Is their membrane permeable? How would that all work? It's close. The answer is with a replication fork, because like that's a utensil and then it's like a joke. All right, just gotta make sure you're with me. Gotta make sure you are with me. See you on the other side. All right. Okay, moving on. And yeah, feel free to use that with all the friends. That's what we're here for. Dad. Okay, I am not a dad, but I do tell dad jokes. Okay, I'm not gonna fight it. All right, fine. There it is. Moving on to the cell division, but also shout outs to Carlos. That was a legit good answer. The septum, right? So we talked about, oh, sorry. I think my internet connection went down for a second. Apologies. So septum formation is important because this is what helps facilitate the division of cells, right? And so as the septum grows inward, there's a point at which there is constriction and sealing off of the two daughter cells. This is demonstrated here by some electron microscopy images. And you see this sort of gradual but continuous septum formation process until you have full division of the two cells. And this has important implications for the microbes and their morphology. You can think of the streptococci that form these parallel planes. Sorry, they don't form parallel planes. They form these long chains, but this is the result of septum formation on parallel planes. So you have a chain of cells, and when septums are forming in these parallel planes, that is the morphology that you would expect to generate. You can have random planes, which results in these more grape-like structures shown here as illustrated by staphylococci. And then you can also have perpendicular planes, which I think are pretty cool. So that's basically akin to a tetrad formation or almost a Rubik's Cube vibe, such as the Sarkinae that are shown here. And all of these morphologies are able to illustrate that how your septation process is oriented will dictate your overall colony formation. We're now going to do a deeper iClicker question here. This one is modeled more off of what you will see on the exams, although even the other iClicker questions will be helpful in terms of preparing you for exam prep. However, my exams definitely feature a fair amount of graphing or an analysis of graphs, because I think, as I discussed on the first day of class, that is really important for how we assess critical thinking and apply critical thinking, right? That's the evaluation of data or the creation of your own graphs. And so I will walk us through the question here, and then we will tackle it together. Whoops, sorry about that. So pretend you are a graduate student in lab studying the effect of antibiotics on bacteria. You have 4 tubes of the same bacteria, and in each tube you add a different compound, W, X, Y, or Z. After 1 hour, you measure 2 different activities. You measure RNA polymerase and the percent of peptide cross bridges that have been formed in the peptidoglycan in each of these 4 tubes, and you get the results below. So you went to lab, you put on all your COVID-19 safety gear, and you perform this experiment. Below are your results. And the question you're trying to answer is, what is most likely the identity of compound Y? Okay, so go ahead and analyze this data. Got a bunch of great data here, and then think about what the most likely identity of the compound, the identity that is most likely to be compound Y based on the ones presented here. I can give everyone 2 minutes to work on this, and let me know if you have any questions. All right, 10 seconds left. So go ahead and get those answers in. And no worries if you're not sure, we will spend some time making sure we understand where we're at. All right, let's see what we have. All right, quite a diversity of options. Ooh, this is harder than normal because I unfortunately did not point out a couple of things that I was supposed to, but apologies, we will go through them nonetheless. We have 46% saying penicillin, 11% erythromycin, or sorry, 11 people saying erythromycin, and then 10 and 14 for vancomycin and rifampin. Sorry, I'm a little mad at myself. I did miss a key talking point here. I'm going to go ahead and just walk us through this because I did not talk about what inhibits RNA polymerase activity, which is definitely central to this question. But this will still be, I think, a useful analysis for us nonetheless. So the correct answer was rifampin, and that is one of the antibiotics I was going to talk about, but did not highlight here. So if we look at this slide, you'll see that there are actually a number of additional antibiotics that come into play here. Rifampin is an antibiotic that blocks RNA synthesis. So this is important for targeting bacteria, but it is independent of the antibiotics that disrupt the peptidoglycan cross-linking of glycan strands, right? Here, this is specifically targeting RNA polymerase. The second antibiotic that is included here is erythromycin, and this blocks the peptide elongation by binding the 50S subunit. And so these are two different antibiotics that we can use to kill microbes. However, their mechanisms of action are different, right? So they're not going to affect the level of peptidoglycan cross-linking. If we go on and look at this slide here, we again are returning to what we did spend time discussing, which is the role of the transpeptidase and how penicillin can inhibit that. Conversely, vancomycin is blocking the D-alanine separation, right? So if we then return to this question and look at the data, we can use this information to analyze the graph, right? So first up, we have percent of RNA polymerase activity. We know now, we should have known then, but we know now that only one of these four antibiotics is going to directly block RNA polymerase activity, right? And that would be rifampin. Rifampin is blocking RNA polymerase activity. We see a decrease in RNA polymerase activity with compound Y. Next, we can look at the percent of peptide cross bridges that formed. What are the antibiotics that we are aware of that can prevent cross bridge formation? And you can just say it or type it in the chat. Right, so penicillin and vancomycin, right? So we just re-reviewed that, and we have the penicillin blocking cross-bridge formation. We have the vancomycin blocking cross-bridge formation. And so those could be possible candidates for X, but the question is asking us about Y. So part of the importance in terms of evaluating data is deciding what data is relevant and what data is not. Our world is filled with data. Papers are filled with data. Newspapers are filled with data. Well, not that there are too many newspapers, but there's a lot of data in our world. And one of the things that I hope you'll become more and more confident in doing is figuring out what data is relevant and what trends in the data are important to focus on. For our purposes, because there's no difference in Y, W, and Z, it's hard for us to discriminate any information about Y other than it's likely not penicillin and vancomycin, because if it was, then we'd expect some decrease in cross-bridge formation, which is what we see with X. There is a difference here with Y, and that likely connects it to rifampin because you have decreased RNA polymerase activity. So I encourage you to take these questions into your notes, to play around with them, ask what would it look like if you were to graph a treatment with rifampin instead of look at the data, right? You could very easily flip this question on its head and say, oh, like I'm supposed to graph this question with what rifampin treatment would look like and what penicillin treatment would look like, only given this axis and this axis, right? And then you would need to use your knowledge of the microbiology to figure out the trends. One other point I will illustrate or share is that when students encounter these kinds of problems, they're often very hung up on exactly where the bar should be for a graph. So should it be at 50%? Should it be at 25%? Should it be at 0%? And I will say this now, and I'll say this many times during the class, that the exact number is not really important for most of these questions. It's really more about the trends that you see. So the relationship from one graph or one bar to another bar. If you're treating with rifampin, it should be lower than these other conditions relative to each other, right? Whether it's at 50, 25, or 0 doesn't really matter, because at a certain point, it's also about the dose of rifampin that you're giving, the length of exposure, maybe the temperature. There's a whole range of factors that could impact the effect of rifampin. But the key takeaway is the relative trend or difference from one sample to the other. So we will continue to do various iterations of these problems and others. Hopefully I'll remember to talk about rifampin. Sorry, that was my fault. I apologize. But this is something that I hope will alleviate any stress you have about the content we cover, because I will try to base test questions off of the ones we go over in class together. Are there any questions about this strategy for asking questions or how you answer the questions? All right, so there is a question in the chat. Was this question possible to answer without memorizing what each of those antibiotics does or do? So I would say that you don't necessarily need to memorize what antibiotics do, but because it is going to be an open book exam. However, if we are spending this much time talking about penicillin, vancomycin, rifampin, and erythromycin, then I would interpret that as a sign that it would be good to be familiar with what they do. So I'm not going to tell you explicitly what is on the test, but if we spend 10 to 15 minutes talking about 4 antibiotics, then I have to test you on something. I'm not going to test you on something that we haven't talked about. So, you know, use your evaluation skills, see what you come up with. So there you go. Any other questions before we get to a few more slides? He's going to. I'm not going to bamboozle you. I'm not even sure what that means or how I do that. So if the question asks about compound X, then the answer would be vancomycin or penicillin. Right, exactly. So if this was about question X or compound X, then it would likely be vancomycin or penicillin. And you could think of other iterations. What would this look like if compound or a test or what would it look like if a tube was treated with both of them, right? So there's like all sorts of fun things you can do. But that's the general idea. Okay, so let us move on. So right now, we're going to talk about polarity and aging. Yes, aging happens to humans. It also happens to cells. And polar aging is when you have poles, so two different ends basically of a cell with different um, origin and age. And this actually has a lot of interesting consequences for microbes. You could think about it as a ship. If one end of your ship is brand new and the other end of the ship has been there for 20 sea voyages, I know what end of the ship I would be standing on, right? And it's not the old one. So this has implications for bacterial cell differentiation. This includes how daughter cells can be generated for the flagellum versus stalk transition of Caulobacter crescentus. And a number of different molecular studies and genetic analysis have looked at specific proteins that are players for determining where there will be a new pole that is formed, as well as the placement of the flagella. This is illustrated in a little more detail here with the coordination of multiple proteins. You have a swarmer cell that will lose its flagella for reasons that aren't entirely clear, but there is flagellar loss and a stalk will grow in its place. You then have the presence of tip N, which is the protein that indicates the position of a new pole, as well as the protein pod J, which moves to be at the pole with tip N. And at the same time, you have div J being produced at the end where a stalk has formed. So you have div J production as well as pod J and tip N being at the same place together. The flagellum then will grow at the pod J end as there is delocalization of tip N across the cell. And this then helps to coordinate division of the cell. And you have this stalk cell as well as a flagellar cell or flagellated cell. And this pre-divisional cell is still intact until you have this tip N localization at the septum, right? So it used to be delocalized all over the place. It meets in the septum, and then you have a new pole as well as old poles on both cells after you have the complete division of the original mother cell. And then this process can begin anew. So it sort of goes with the song, make new friends, but keep the pole, if you will. But yeah, basically you have this cycle mediated by these three proteins to give a difference in terms of polar aging, as well as stalk or flagella formation. We're going to stop there for today. And please let me know if you have any questions. I can stick around for a couple of minutes. I hope everyone has a great weekend. Practice your dad jokes, and I will see you on Tuesday. Take care.
Dr. Adams?
Hello.
Hi, are you